I'm Still Rosello.
I'm Valerie Wendell.
And this is lesson 12, group problems. So we're just going to alternate 1, 2, 3, 4, and 5. And Valerie's going to start us off with 1.
All right. So for number 1, it says the transcriptional regulation in bacteria involves a combination of regulatory proteins, DNA sequences, and small effector molecules. Note the list of terms below, and it gives us a list. Then for A, it says which of the terms describes or is an example of regulatory protein. We got activator and repressor. For B, which of the terms describes or is an example of a small effector molecule? And we got an inducer, allolactose, cyclic AMP, and an inhibitor. For C, which of the terms describes or is an example of a sequence found within the DNA? And we got cap, lac Z, lac A, and then +1 and -10 sites there. And D, which of the above is not involved in the lac operon? And so we took this, so there's like 2 ways that you could take it. It's either like none of them, because technically they all are involved in the lac operon in some way, or it could be that she's specifically asking like not the operon, and so what is not involved is the activator, the inhibitor, the allolactose, the C-amp repressor, or the inducer. Those are all like effector molecules or different proteins or enzymes that come in and affect the operon. And then for E, it says which of the above is an example of structural gene, and we got lac Z and lac A. Yeah, sorry.
Okay, so for 2, fill in the blanks. For A, the lac operon is regulated by 2 inducers, allolactose and cyclic AMP. The binding site for sigma factor in RNA polymerase is called LACP, while the CAP site or cap site is the binding site for the activator that regulates the lac operon. For C, the enzyme adenyl cyclase converts ATP to cyclic AMP. For D, the -35, -10, and +1 sequences are found within the LACP portion of the lac operon. For E, the LAC-I gene encodes the lac repressor, while the LAC-Y gene encodes lactose permease. For F, attenuation can regulate the gene expression in bacteria by causing transcription to terminate prematurely. And for G, an E. coli strain that contains 2 copies of the lac operon is called a merozygote strain.
Okay, so then for 3, it says, suppose you are examining the lac operon in E. coli. In your experiment, the activity of the enzyme beta-galactosidase is measured by including xGal and IPTG in the growth media. IPTG can mimic allolactose. IPTG can bind to the lac repressor and release the repressor from the operator. xGal is a molecule that is a substrate of beta-galactosidase and turns the E. coli cells blue when metabolized by beta-galactosidase. So parts A through E below describe 5 different cultures conditions that will be used in your experiment. Determine if the culture conditions described in parts A through E will result in blue E. coli cells. And then for this problem, assume that your E. coli cell contains wild-type lac operon components, that allolactose is absent in all cases, and that abundant ATP is present in all cases. So I'm going to tell you what we got, and then I'm going to describe how we got what we got. So we got that none of them will be blue except for B, and here's why. There is one condition that you need to be able to have it transcribed and made blue, and that's if you have glucose absent, because if glucose is present, then it shuts down the operon and it won't work. So you have to have glucose absent. IPTG has to be present so that it can bind to the repressor and release it from the operon. And then xGal also has to be present, because when xGal is combined with the beta-galactosidase, that is what makes it blue. So when you have no glucose, IPTG is present, and xGal is present, then you'll get blue. Anything else will make it not blue. And so that is why all of the others are not blue, because they don't have all 3 of those. And that's it.
That's it, yeah. Okay, so for number 4, it says, below parts A through E are the lac operon genotypes of several E. coli strains. Please note the following, so on and so forth, what it reads there. And just asking, do these E. coli strains display constitutive, inducible, or non-inducible expression of the LAC-Z gene? And for the heterozygote strains, parts C through E describe the overall phenotype of the cell, do not provide separate answers for each copy of the lac operon in the cell. So, 4 and 5 kind of run along the same lines as far as understanding what the minus or the plus or the S or the C is really doing to the cell that changes what type of what type of display it's going to have. So constitutive means it's just going to always run no matter what. If you have lactose, which is going to give you allolactose, which is an activator, or if it's inducible, so it's only active in the presence of an activator, or if it's non-inducible, which means it's never going to run no matter what you do to it, no matter how much stuff you add to it, it's just never going to happen. So I'm going to just kind of give you the answers of what we got on some of these and maybe go through why. But for A, we got inducible. For B, we got non-inducible. For C, we got constitutive. For D, we got non-inducible. And for E, we got constitutive. So the way it kind of breaks down on these ones, so if you go through, the Z is a Z plus on all of them, so as long as the other ones are going to go through, it's gonna produce what it needs to. So if you look at the I, for example, for A, if you're looking at the I, so if it's an I plus, that means the repressor is made. If it's a P plus, that means that the polymerase is active, so the RNA polymerase, and it will run through. And the O plus means that the repressor will bind. So for this one, inducible, since we have repressor and it will bind and the polymerase will go through as long as the repressor is able to be taken off, then if we introduce lactose or allolactose as the activator that will bind to the repressor and remove that, then the polymerase can go ahead and do its job, and you will be able to induce it in the E. coli strain, so you'll get what you want, whatever gene that might be. So for B, we have an IS, which is a repressor is going to be made, but it's not going to be able to be activated by allolactose. So in this case, the repressor is, we can just call it a mutant repressor. The mutant repressor will be made. It will bind to the site, and polymerase will do its job if the repressor can get off. But since the repressor is mutant, and it will not be able to be removed by an activator of allolactose, then it's never going to produce, no matter what you do to it. So that's why that one was non-inducible. And then for C, it's a little bit different because you've got two parts to it. It's the heterozygote form, so you're going to have to take each of them into consideration individually and then also check to see if one is going to affect the other. So for C, we got constitutive, which means it's always going to run. So we have repressor, it will bind, and the P on this first part of it is not active. So this one, no matter what you do to it, it's not going to make anything. But this one right here, in the F plasmid, we have repressor will be made, the polymerase is active, and the OC means that the repressor cannot bind. So in the case where the repressor cannot bind and you have an active polymerase, it's going to go ahead and start making things no matter what. So if there's an activator, if there's not an activator, it's going to go ahead and start producing. So in this case, even though this one's not making anything, it's still going to be making stuff from the F plasmid. And we did the same thing for D and E, just going through that way. I'm not going to explain all of them, but you can figure it out for yourself on that one. So we'll go ahead and move on to 5 and maybe explain a little bit more on how we got that.
Okay, so we've got it all drawn out here, And so this is what we have been given, and then we need, well, I'll read it to you. How about that? So the question concerns the Jacob Monod and Pardee experiment that utilizes the mutant and merazygote strains of E. coli to examine the lac operon. Suppose Jacob Monod and Pardee use the strains listed in parts A through E below in their experiments. Recall that each experiment involves splitting the E. coli culture, like example, each strain listed in parts A through E, into 2 tubes. One tube does not contain lactose, so minus lactose, and while lactose is added to the other tube, so plus lactose. Your job is to figure, this problem is to figure out how much yellow product will be present in each tube at the conclusion of the experiment. And so we'll use the following scales. 0 indicates no yellow color, 1 indicates the amount of yellow product when 1 copy of the lac operon is transcribed, and 2 indicates the amount of yellow product produced when 2 copies of the lac operon are transcribed. And so for A, we have all of the wild type except for this I, which is what makes the repressor, and right, that makes the repressor. Yeah, and so anyway, so the repressor will not be made. And so, because of that, then it should.
So the repressor is not made. The repressor can bind, but since there's no repressor, it won't ever bind. But your P site's not active. So because the P site's not active, the polymerase can't ever produce any genes. So basically, it's like a defective operon. So that's why in the presence or not in the presence of activator, it's never going to make anything.
Right.
Yeah.
Yes. Okay, and so then in B, you have the repressor made, and but you have the operating, the site where the repressor binds, and it's not able to bind there, and so it won't bind. And so with lactose present, you will get yellow, but without lactose, you won't get yellow. Does that seem up to you?
I actually think you might get yellow.
I think you will get yellow, yeah.
Okay, we're changing our answer on this one after looking at this. You will get, you should get a yellow coloration right here.
Because the repressor cannot bind.
Okay.
And so
This just proves that you learn when you teach, I guess.
Yeah.
So, okay, so for C, let's hope we get this one right too. There have been some questionable results from different groups that we've looked into, so hopefully this is right. But for part C, you basically have 2 operons, and the deeper coloration, the yellow 1 versus yellow 2, is if both of them are working, you're producing twice as much, and it gives you that deeper yellow color. For this one, lactose, we got each of them were producing, and in the absence of an activator, only one of them was producing. So repressor was made, repressor did bind, and it is able to go through in the presence of an activator, which there comes one from there. And on this one right here, oh man, I think we might have messed this one up too, but the repressor is made, the repressor binds, the P is not active, so it shouldn't go through on this one. Interesting. Is that right? Did you write it out wrong?
I did. It's supposed to be a
OC.
let's see. Yeah.
Okay. I see what's happening here. Did we write B wrong? Or did we get that one okay? Plus C plus. No, that one's good. Okay. So maybe we were not wrong on this one. But you have the repressors made, repressor does not bind, and the P's active, so it's going to go ahead and run through. So that one's correct for 2 right there. And then lactose, since this one needs an activator to run through, and without lactose you don't have an activator, this one's just going to run through since there's no repressor that can bind here. It's just going to go ahead and pump something out. So that's, we have yellow 1 on there. And for D, check it here. Minus plus plus, plus plus, minus. Perfect. Okay, so for this one, no repressor is made, repressor can bind, and the P site is active. So this one, it's a little bit funky because even though repressor is not made by the chromosome, the repressor is made by the active site here. So this repressor will bind to this site right here, and so you need to have an activator for this one to go ahead and run through. This one doesn't have an active P, so it's not going to run through no matter what you do to it. So this one will be activated in the presence of a activator, which is here, and it will not be activated without it, so 0 on this one. And then this last one, no repressor. Repressor can bind, the P site's active. We have a mutant repressor that will actually bind to both sites on this one here, and so since the mutant repressor is binding to both of them, the activator is never going to be able to take that off. And so even though both of the P sites are working, no matter what you do to it, it's not going to run since that repressor is always going to be there.
And that's it. Thank you.